accommodating me for not being there in person today. My apologies for not being present. So I wanted to introduce myself. I am the Director of Multi-Omics for Bruker. This is actually a new role for me. I just got this position within the last couple of months. I was previously driving our market here in the Americas, and apparently I was doing a good job. So they've put me in a more global position, which is wonderful. I get to now experience research all over the world. And I'm really excited to share with you guys what our portfolio is for doing 4D-Multiomics, which mostly includes at this point Metabolomics and Lipidomics. And for the benefit of people who are not as familiar with Metabolomics and Lipidomics as they are with Proteomics, I did want to take you through some of the background of how we do Metabolomics and Proteomics in a general context. So Metabolomics in a systems biology context is presented through the central dogma of biology, of course, starting with genes, going to proteins, which of course is looked at through proteomics, which then can be translated into our metabolites, interacting with our proteins, and can give us information about upstream biological processes. Now, this is involved with a lot of molecular biology research. We can be looking at different human samples, mouse models, cell cultures, and even things like plants. This is also very relevant to things that are involved with any kind of food or nutrition, medicines, environmental exposures. So looking at exposomics, this has applications in pharma, DMPK, and any kind of applied situation where we're looking at this in a nutritional context, people that grow plants and agriscience. It's a very broad topic when we talk about metabolomics. And of course, this will give us, if we're looking at it with a mass spectrometry solution, we're trying to capture as much of this metabolome as possible in order to give us some kind of fingerprint that's going to give us a profile of our overall phenotype. Now, this phenotype that we're looking at is going to be relevant to what we can see in our mass spectrometer. And not only are we going to see what is relevant to our whole body or our system as a whole, but if you're looking at this in a human context, there is also implications with the microbiome, which we can also capture with these kinds of experiments. And then not only in a single snapshot, we can also look at this through over time with a context to aging or any kind of dosing over time experiment. And this can also be captured in things like metabolic flux or longitudinal studies. Now, when we come to what metabolomics is and what our ultimate goals are for metabolomics experiments, we really want to get a comprehensive solution to be able to look at the entire metabolome. We want to be able to quantify, and this is something that we want absolute quantification. We want a complete picture of our metabolome. And this goes back to some of our this is very similar to genomics, where we try and cover with full genome sequencing our entire genome. And in a systems biology context, the same thing applies to both proteome and metabolome, where we want to define an entire complement of small molecules within a biological matrix of interest. Now, ultimately, we can't do this in one single experiment. We tend to move towards things like targeted quantitation. So if you have an idea of what your analytes of interest are, this is where you would do a targeted type of metabolomics experiment, where you would look at somewhere between 10 and 150 targets, and you can quantify those based on calibration curves with analytical standards. So this is our hypothesis-driven research and monitoring, and this allows us to look at single sample analyses in a more clinical type context, where we're looking at changes that are absolute. Now, when we talk about global profiling, this is where we're taking a more unbiased look at our overall metabolome, and we're trying to capture hundreds to thousands of metabolites in a single experiment. And this type of quantitation tends to be more relative. And the purpose here is to do this biomarker discovery or overall metabolome fingerprinting. Now, our solution for this is our timsTOF Pro 2. This is our workhorse that we use for 4D-Omics workflows. It provides us with enhanced measurement specificity. We use MoMA, or mobility offset mass aligned, which is something that Shorjo talked a lot about. And this provides us with clean, unambiguous MS data to allow us to annotate those metabolites. And that also gives us the capability of matching against CCS values, which is a very important part of having accurate annotation for those small molecules. And one of the reasons why these are very hard to capture and annotate appropriately is because the chemical diversity within our biological space is so diverse that it can be very complex to try and accurately annotate those on MS alone. So when we look at a typical 3D experiment, we are looking at the fundamental unit being the peak. And this we can call a peak, we can call it a feature, whatever you want to call it. It is a single compound that we are trying to annotate within thousands of compounds. And this becomes a very big data problem where we're trying to look at our M to Z, our retention time, and our signal intensity. So our physical properties of these chemicals that we really want to capture is the M to Z, and this also will include our MS fragmentation pattern, which we can utilize in annotation, and our isotope distribution, which is going to give us an indication of our chemical formula. And in addition to that, we can also use retention time in a chromatographic gradient. This is specific to the behavior of the chemical within a chromatographic stationary phase. And this does tend to be a little bit more variable in experiments, and gradients should be set and standardized and optimized so we can get consistent results over time. But anyone who's familiar with liquid chromatography knows that we do tend to see retention time drift. We get batch effects of columns that will cause slight changes in retention time. So it tends to be more of a variable than we would like. We would like this to be more static. And then the thing that we're really interested in is our intensity, because this is going to give us that indication of how that molecule is behaving within a metabolic pathway. Now, the ones that I want to focus on are these physical properties, and M to Z becomes very, very intrinsic in our abilities to annotate and give us indication of, with that mass accuracy, what that analyte is and how it's changing within a biological experiment. In addition to this, with a TIM system, we now are adding collisional cross-section. And this is what provides us with our fourth dimension and giving us 4D omics. So let's take a closer look at a close-up of one of these peaks that we're measuring. And specificity really is the key. We want to be very specific on what this analyte is, where it's eluting, what its intensity is. But there is also isobaric and isomeric species. So isobaric species being compounds that elute within a very close mass that are within 1 Dalton that couldn't be separated by a quadrupole to give you pure isolation of that compound. Quadrupoles are typically limited to about 1 Dalton. When you isolate in a quad, you tend to reduce your signal intensity overall, and this becomes problematic for keeping up with that sensitivity while you're trying to achieve that specificity. So how do we deal with the fact that there might be more than one biologically distinct species underneath that one peak? And this is where this 4D workflow really comes in handy. So just to give you a recap of PASEF, or parallel accumulation and serial fragmentation, that Shorjo went over in the previous presentation, the key here really is having this mobility offset mass alignment of our MS fragments with our initial precursor. So we do have alignment between our initial species that we're collecting in an MS1 experiment, and then associating that with the MS fragmentation pattern. in a experiment where we're trying to associate these, the ones that I want to point out are the two lighter red ones in the middle that have co-eluted chromatographically, and they are isobaric, so they have the same M to Z value. And when we look at them in an MS-MS fragmentation pattern, if we did not have this additional axes or dimension of mobility separation, these would co-elute and be convoluted. Now, when we look at this in a big experiment where we have many peaks eluting simultaneously, we might have multiple co-eluting species within our metabolomics experiment. And TIMS allows us to be able to separate those and give us those clean MS-MS spectra that are going to be attached and associated with our MS1 that are now separated within the ion mobility dimension, as well as our mass analyzer dimension, so M to Z. Let me give you a small example of where this is beneficial. This is an example of vitamin D epimers that are being separated within a TIMS cartridge. And this allows us to take things that are isobaric, only changing by a shape of our molecule. And this allows us to do separation within that ion mobility region, using that fourth dimension to separate those two epimers and give us two completely distinct biologically active forms of that metabolite, which we can then further do not only a CCS collection, but MS-MS fragmentation to give us those nice, clean MS-MS spectra to say, yes, this is the analyte that I'm actually interested in, and I would like to do quantitation, whether it be a relative quant in a global profiling experiment or within a targeted experiment. So this is an example of what an untargeted 4D experiment would look like. This is actually an example of SRM 1950 plasma, which we've done an extraction on. This is a lipidomics experiment. It's something that we would like to run quickly. If we have tens to hundreds to thousands of samples, we want to be able to have the most efficient runtime as possible. So this experiment is a 5-minute experiment, and you can see the standard base peak chromatogram on the bottom frame here, and the top frame represents all of the MS-MS data that we've collected from a single 5-minute lipidomics experiment. So each one of these red diamonds represents a precursor containing a mobility selected and mass selected clean MS-MS spectrum. So I'll bring to your attention in the total number of MS-MS spectra that we've collected. When we run with a TIMS PASEF experiment, we have over 25,000 MS-MS spectra that are being collected. If you ran that in a comparable 3D experiment without ion mobility, we're looking at about 3,800 MS-MS spectra that are being collected. And that's a significant increase in the number of spectra that we're collecting. That's going to allow you to more efficiently and accurately annotate your analytes of interest. I will also bring to note that the number of precursors that are targeted is going to be less than the number of MS-MS spectra that are collected. And I'll go through that in a moment as to why that is. This allows us to have increased annotation, be it on our different types of annotation workflows. And then we can also look at we can use multiple different workflows to annotate to give us combined annotations that are unique. This is a very fast type of experiment. So on average for metabolomics and lipidomics experiments, when we're looking at MS-MS speed, we're talking about over 100 hertz of MS-MS data collection. And this allows us to get over 75% of MS-MS coverage within our samples. Okay, so the PASEF data acquisition that we're talking about here involves a number of cycles. So when I talk about how we collect MS1 data, and then on the next round of TIMS, we would collect MS2 data that's associated with that precursor, like a standard DDA type of experiment, except with that fourth dimension. Now, with metabolomics and lipidomics, we actually do 2 cycles of MS-MS. And this allows us to get about 4 orders of magnitude of MS-MS coverage. So our analytes that are eluting, that are of high abundance, are going to be collected only once because they will have good fragmentation patterns and good signal intensity. But there's going to be some down here in the weeds that anyone that's familiar with metabolomics and lipidomics type of experiments, they might be of a lower signal intensity. And if you collect that data only once, then you're not going to have necessarily the best quality of MSMS spectrum. You might only have your precursor there and nothing left over. So the scan speed adjusts to have more ions come into the system, which then gives you a higher signal intensity. And then the collision energy is also adjusted as well to give you more fragmentation and give you more peaks in your fragmentation pattern to be able to do annotation. And this is how we can get over 75% of our features containing MSMS. Now, I've talked a lot about how MoMA allows you to get those nice, clean MSMS spectra, but what about that CCS value or collisional cross-section? So CCS is something that we are really promoting to use as an annotation tool. And we're not alone in this. Anyone that has been using ion mobility, especially in the small molecule space, has been using CCS to annotate their analytes or as an additional annotation tool. So one thing that's very important with our instrumentation is reproducibility. So this was actually coming from a collaborative experiment between our Bremen facility and the university in Munich that both have timsTOF Pro 2 systems. And this shows the reproducibility of our CCS values between systems. And I'll make a note here that the Bremen instruments used analytical flow and the Munich instruments used nanoflow. And regardless of how you're ionizing, you are still going to get reproducible CCS values. And this goes the same for MALDI as well. So this is ionization source independent information that's going to provide you an additional annotation tool. And when we're looking at correlation coefficients, we can see that even the lowest correlation that we have between CCS values between instruments is at 0.991. So this is a very high reproducibility of our CCS values. Not only are we able to reproduce this in positive mode data, and as the last example was a lipidomics experiment, we can also do this with negative mode data and of course with small molecules as well. So when you're looking at positive mode data, they are very reproducible between systems. And this was a collaboration between Professor Jeremy Nicholson's group at the Australian National Phenome Center and again, our group in Bremen. And this is showing both positive mode data and negative mode data are within less than, well, definitely less than 1% of deviation between CCS values across platforms. Now, one thing that we like to talk about as well is other ion mobility systems. So the gold standard of ion mobility and the initial system that was developed is the drift tube system. And the drift tube, I will say, is a much more complex system. It has a much bigger footprint with respect to the size of the drift tube. The longer the drift tube, the better resolution you have. This is not the case in a TIMS cartridge. A TIMS cartridge is about 10 centimeters long. So we can really optimize the size and shape and the separation within a much smaller space, but we can still be comparable to drift tube values. So if anyone is familiar with John McLean's group at Vanderbilt University, they've developed this CCS compendium, which has about 5,000 compounds in it of small molecules. And we are very reproducible, but again, much less than 1% of deviation between drift tube values and TIMS values. Now, chemical space, very diverse and very big. So we're talking way more than 5,000 compounds. This is where we start implementing things like machine learning models to predict values instead of using experimental values. Obviously, I would always say the experimental value is going to be more reliable and robust. However, when you don't have that information in an experimental database and you want to be able to still utilize that CCS value, a machine learning model is very accurate at giving you a predicted value that's going to match with your experimental value based on the size and shape of that molecule. And right now, CCS predictors report about a 3 to 4% error. This is actually being reduced. I would say that it's about 2% now of deviation between predictive values as we increase our training sets and develop more intelligent machine learning algorithms and models. I would have to give some kudos to Professor Shenqiang Xu. He's one of the first ones to utilize predictive models in CCS, and we've worked a lot with him. He has 2 of our timsTOF Pro 2 instruments, and we've taken those machine learning algorithms and built upon them and implemented them within our data analysis workflows. And this is what we call CCS Predict Pro. Now, let me give you an example of how we can use this in a real experiment, in a real life example. So this is, again, a lipidomics experiment where we're trying to implement a shorter runtime. This time we're looking at a 15-minute runtime instead of a 5-minute runtime. However, we still experience co-eluting species. So we have a PC or a phosphocholine and a PE or a phosphoethanolamine that are isobaric in mass. So they are only 36 millidaltons apart. They will co-elute chromatographically. Theoretically, if you run a much longer gradient, you would be able to separate them, but that kind of defeats the purpose of trying to shorten your runtimes and increase your sample throughput. Therefore, if we implement TIMS, we can take this co-eluting species, and instead of isolating in a quad and co-eluting and having a convoluted MSMS spectrum, we can separate those in ion mobility. And as you can see down here with the mobility resolution, 36 millidaltons is enough to allow us to have separation within the TIMS cartridge. And we can get 2 distinct CCS values. These can be used in annotation workflows. And we can also have those nice, clean MSMS spectra using MoMA. Slightly more complex example, like I said, you could theoretically separate those 2 PEs and PCs by chromatography. In some cases, that's not the case. When the head group is the same, it's much more challenging to separate them chromatographically. And this is an example where we were able to separate structural isomers of exactly the same mass of these 2 phosphocholine. And despite the fact that they are isomeric, the double bond positions actually kink the structures significantly enough to give them separation in our ion mobility cartridge. And this gives us a high resolution separation of isomeric lipids. Not only can we do this for untargeted workflows and overall profiling, giving us separation and annotation tools, but we can also do this in quantitative workflows. So this was an example moving from lipids back to small molecules, just to show the breadth of the capabilities of the instrument. This is a pharmaceutical spike within urine, and we did a gradient or a concentration dilution series to show a calibration curve and how reproducible the data was over a very low concentration. So we can see that we have a really nice high dynamic range here. And when we're looking at the low concentrations, what I want to point out is that the MSMS spectrum looks exactly the same. We're very reproducible within those MSMS data because we're able to separate these within the ion mobility space. Why this was such an important example is because you often have co-eluting species that are unanticipated, whether it be from your sample matrix or from your solvents or from any kind of plastics that you're using within your sample preparation protocols. Sometimes there's just no way of getting around it. And if you want to quantify something at a low level, you want to make sure that you're looking at only that species of interest. So this was an example of pyridophin at the 1 ppb spike level, and it is co-eluting with an isotopolog of a contaminant within that mixture. And in a normal 3D experiment, these 2 would not be separable within the quadrupole. They're too close in mass. They're only about 100 millidaltons apart. But when we employ ion mobility, these 2 are separated, and then you can get 2 clean you can get 2 different MSMS spectra that are going to be more accurately matched to your database spectrum. And this is going to allow you to have more confident low-level analysis when you're doing a targeted quantitation workflow. Now, when I'm talking about low levels, we also want to talk about how we can increase our signal sensitivity. So one of the newer things that we've been employing within our metabolomics and lipidomics workflows is the VIP HESI ionization source. So this is a vacuum insulated probe heated electrospray ionization source. We have these for both our QTOF and timsTOF series of instruments. So the secret to this type of ionization source is that this is the actual vacuum insulated part of the probe. So the center part where the needle is actually going through the probe itself is going to remain nice and cool. And when it reaches the tip of the needle, it's going to be cold right until it hits that blast of gas and heat, which is going to promote more efficient desolvation of your analytes. It will increase your ionization efficiency, and because it stays cool within the jacket, it prevents the degradation of any labile molecules. So it prevents in-source fragmentation. Now, just to show you an example of how this works and what it actually looks like with real data, this is our standard electrospray ionization source and our compared with our VIP HESI source. We tend to see about 5 times greater sensitivity compared to a normal ESI source. And this does give us triple quad-like sensitivity when we're running these types of experiments. And this is actually a technology that was developed for our triple quad systems initially, and has since been revamped and put onto our timsTOF and QTOF series of instruments. Now, it's not only good in positive mode, but it also works quite well in negative mode. And in metabolomics and lipidomics experiments, we do tend to see less signal intensity in a negative mode experiment than you would in a positive mode experiment. And when you employ a VIP HESI source within your experimental workflow, you can get down to quite low levels and still see a very nice, significant increase in your signal sensitivity. Again, I have to promote SRM 1950. We use this as a lot of our baseline testing to ensure that our instruments are robust and efficient and reproducible. So that kind of brings me to the end of the hardware part of the talk. And I know there is a lot of interest in single cell work. I will say that our workflows for single cell are not quite as robust and worked out as the proteomics business unit. However, I do have some slides at the end to show you what our capabilities and how we've gone so far. But first, I'm going to talk a little bit more about software and how we do data analysis. And I want to introduce to you Metaboscape. This is our all-in-one solution for doing data analysis in untargeted workflows. So this takes all of your MSMS data that was acquired using PASEF. It is also applicable to 3D methods and flow injection analysis methods as well. This allows you to do things like peak picking, annotation, and statistical analysis. So it is compatible with all of our different mass specs. So we can use it with our timsTOF systems. You can use it with an MRMS system. You can use it with our SCP. And of course, you can use it with a standard QTOF system as well. So the data is all compatible with, oh, and I should also mention, it is used a lot in MALDI imaging too. And the way that the interface works is once you do your sample processing, you will have a sample table that shows you all of your different sample cohorts. So this is where you would set up things like a wild type versus knockout, a time series, or a dosing experiment, or any other type of sample cohort that you want to set up for doing your statistical analysis downstream. We then have a feature table. This is where all of our analytes are selected, or our peaks are selected. I will mention that each one of these is what we call bucketed. So including all of the different adducts, they are formed into one single feature within the table. Every time that you click on a feature, you do get a graphics view of the signal intensities between your sample cohorts. And then we can also easily look at our statistical analysis that has been employed within this one single screen. And of course, there's many other tools within Metaboscape, and I'm going to take you through a lot of those in a moment. But I also wanted to discuss how we actually get this feature table, because this is an important part of how you do metabolomics experiments. And and I perhaps should have mentioned before a little bit of my background. I did a postdoc in Gary Shustak's lab at the Scripps Research Institute in La Jolla, and I spent a lot of time working on XCMS. I've also spent a lot of time using GNPS, developed in Peter Dorstein's lab, and they're awesome tools. They're open source tools and they are freely available, and they do a very good job. However, when you're trying to implement this in a more standardized way, that tends to be a very slow way of doing it. And Metaboscape allows you to do this in a much more rapid fashion and a much higher processing speed. So to detect features, we employ an algorithm called TRex 4D, or time-aligned region complete extraction. So this allows you to set the number of points across a chromatographic peak that you would like to consider to be a peak, and a signal intensity threshold. And this is going to take a first pass over all of your M to Z features and set them as a peak, or a peak of interest that is going to be looked at within your feature table. However, in some samples, especially if you're looking at real biological samples, you might have a single sample within your cohort that is less than those intensity thresholds. And we want to be able to go back into the noise and set a lower set of thresholds to say, okay, is this really a zero value, or is it actually there within the noise? So it will recursively extract any features within that noise region, so you don't have to deal with those zero values, because zero values for statistical analysis can be very annoying to deal with. You can always tell which features are recursively extracted. They always show up in your feature table in an italicized way. So when you look at each individual feature within the feature table, we report retention time, CCS value, our measured M to Z value, what the associated monoisotope would be, whether or not you've collected MSMS data with it, and all the different ions that are associated with that particular feature. And that would include things like neutral losses, for example, water, sodium adducts, potassium adducts, and these are optimizable within your data processing tools, and you can set whatever adducts you want. So if you're using different mobile phases that have additives to them, that you would have a various adduct that's attached, you can add that to your feature table just by putting it into your sample processing. You can look at each individual feature to pull up all of the raw data that's associated with that particular feature. So this would include things like all the individual CCS values, all your individual intensities. And I will also note that calibration is done on every single sample. So each one is calibrated for mobility and mass. And calibration is very simple. It is not compound class dependent. So it doesn't matter if it's a small molecule or a lipid or a peptide. We use the same calibration for every single type of molecule. And with each data processing portion, this is in our workflows, we tend to inject a plug of mobility and mass calibrant, and that allows you to keep every single sample within that sub ppm level of accuracy. Now, when we're doing annotation, you now have all of your features. We do have a number of libraries that are available for doing annotation in Metaboscape, one of which is the Metabolo-based personal library. This contains over 100,000 compounds in it with MSMS data, both experimentally derived and in silico derived MSMS data. And this was developed in Gary Shustak's lab at Scripps. So this one is near and dear to my heart, highly associated with the Metlin library. We also have the HMDB metabolite library. This is a more concise endogenous metabolite library, but it is highly curated and also contains retention time information for a standard gradient. We also have the NIST 2020 mass spectral library. This one contains about 30,000 compounds in it, and they're all biologically relevant compounds and includes both endogenous and some pharmaceutical type compounds as well. And it is a very robust library, and it's actually my favorite. I would say it gives you the most bang for your buck when you're doing any kind of annotation workflow. We also have a very small free Metabo-based plant library that was developed in collaboration with Lloyd Sumner's group at the University of Missouri. So when we're doing annotation, you employ your library, you do an annotation of your entire feature table. And now we want to see how good is that match to our experimental data. So we have what we call annotation quality scoring. And this allows us to take our M to Z value, our isotope pattern, and our MSMS data, and be able to match it and give it a score to say how good is that match with our experimental data compared to the library. And of course, these are the ones that are derived from M to Z. And then we also can do this with TIMS derived data with our CCS. And this can be done either from a CCS library that's experimentally derived, or we can use CCS Predict Pro to do an overall annotation based on our predictive algorithms. We can also use retention time if you have a standard gradient and you have those set within your workflow. And then, of course, our final value that we're interested in is that peak intensity. This is the one actual variable that's being measured and the one that we care about. In untargeted workflows, we are looking at a relative quantitation. So we're looking at how this is changing between our sample cohorts. Of course, this isn't used in our annotation quality, but these are the features that allow us to accurately identify that feature so we can get an inference within our biological experiment what those changes are meaning with respect to metabolism. We can also do statistical analysis within Metaboscape, and we have both multivariate and univariate statistics, including PCA, PLSDA. If you're looking at 2 sample cohorts, you can apply a simple t-test to give you your nice little volcano plots as shown in the top left. PCA here is probably one of our more powerful tools, and I would like to say that the way that we do statistical analysis is obviously to all of the standards of statistics. However, it's interactive. And I like the interactive features within our statistical analysis package because it allows you to be able to click on a feature within, say, the loadings plot of your PCA plot and send you right back to that feature of interest. So be it a known or an unknown, we can then look at the individual chromatograms, we can look at the mobilograms and the overall intensities that are associated with that one feature that's pulling our PCA components out of alignment. So when we're looking at this so this is all from our annotation side of things. So we have, but what happens when we have unknowns? So for lipids, we have what we call a rule-based lipid annotation workflow. And in addition to the rule-based, we also have the Lipid Blast library. This is in conjunction with Oliver Fien's group at UC Davis. But again, that library is based on experimental data and some in silico data. So we want to be able to have multiple modes of being able to annotate. So this is akin to our predictive algorithms because we're using what we call CCS hyperplanes to be able to predict our CCS values. And then we're also using our rule-based annotations based on the Lipid Standards Initiative to be able to take the chain length, head group, number of double bonds, associating that with CCS and giving us a prediction of what that species would be. Now, something that we employ a lot with lipidomics experiments, and this can be used with other homologous series, but Kendrick mass defect plots are a really good way of both annotating and getting an overall look at coverage of lipids within a single experiment. And for those of you who aren't familiar with KMD plots, this is where you're looking at a series of molecules that are based on changing number of methyl groups within a lipid backbone. So it's looking for those consistent heteroatoms that are your head group, the chain length, and the number of rings and double bonds that are present within your chain. And this is an example of triglyceride series within a lipids experiment. But we can also do this, and I'll just show the chain length is going along the x-axis here with our increasing M to Z. And then with our KMD, that gives us our number of double bond equivalents. So when we look at this as an overview within an entire chromatogram, we can plot M to Z versus retention time and see the overall coverage of lipids just based on lipid classes within our experiment. We can also look at CCS versus M to Z to give us an idea of like where our increasing number of double bonds and increasing chain length are. CCS is going to be represented by the radius of the circle within our plot here. And then we can also look at an actual M to Z versus Kendrick mass defect plot. And this allows us to pull out, this is how we pull out our lipid classes altogether. And then one of the other benefits that we have by using KMD is that we can pinpoint outliers. So sometimes when you're annotating, you end up getting a misannotation based on, it's called it out based on the rule-based annotation or a match to a spectral library. But retention time and CCS dimensions can really give us an idea of whether or not that's been misannotated. So the KMD plot will actually pull out those outliers of things that have been misannotated, and then you can go back and have a further look at what those analytes actually are, or if they're not important to your experiment, they can be removed from your experimental analysis. So we have two different plots that will allow you to detect both our retention time and CCS outliers. Now, for small molecules, we have a slightly different workflow because we don't have the benefit of things like rule-based annotation. And this is where we use our compound identification workflow. This employs 3 different modules within Metaboscape called SMART formula, which generates our chemical formula, compound crawler, which allows us to search databases for putative IDs, and METFRAG, which allows us to do in silico fragmentation. So SMART formula is based on the 7 golden rules, also developed in Oliver Fien's group at UC Davis. And this allows us to look for the best molecular formula that is going to fit our M to Z value, isotope pattern, and not only from the MS1 data, but also from our MS2 data. How it shows up in your feature table, annotations, you'll see an SF for SMART formula. If you look for an annotation library or a spectral library, it would show up as a spectral library annotation. But in this case, we see that this has been annotated with SMART formula, and all the molecular formulas are associated within that. Instead of a name of a compound, you're going to see the molecular formula here. Now, once we get our molecular formulas, we can use compound crawler. This is an online and link to those respective databases and give you a list of putative matches based on that accurate mass and isotope pattern. Once you get a list of putative IDs, this will allow you to go into the METFRAG module and give you an in silico fragmentation pattern of those proposed structures, and then score it compared to your experimental data. So not only does it take an overall score based on your overall spectrum, but you can do a bit of a deeper dive within the spectrum itself and see how well the fragments match in your experimental data to your predicted in silico fragmentation pattern. And of course, I'm going to say that experimental data is going to be your gold standard. I will always recommend an analytical standard to confirm, but this is going to give you a good idea of what that compound is going to be, so you know the direction to go in to say, if I need an analytical standard to be able to do a proper comparison. In silico fragmentation is really good for primary fragments. For those of you familiar with fundamental fragmentation mechanisms and how primary fragments are very easy to form within a heteroatom site. Secondary fragments and rearrangements are very difficult to predict and requires a lot of computational power. So at this point, METFRAG is a really good indicator of primary fragments, but can be a little bit ambiguous when it comes to the other types of fragmentation that can occur within an ionization, within a collision cell in this case. So now that you've had your overall features, many of them annotated, and you get an idea of those relative changes in abundance. We can plot those on metabolic pathways. So we do have a tool within Metaboscape that you can make your metabolic pathway map and plot your changing metabolites on that map. And this is a really nice way to be able to visualize things that are changing within your experiment. So that's our Metaboscape software in a nutshell. We actually do have one more module called Biotransformer, which I haven't touched on here in this talk, but that module will allow you to do biotransformation pathways. So again, looking at a metabolic pathway or a set of metabolic rules or enzymatic rules, and you can take a fragmentation, you can take a molecule and biotransform it, and then compare the fragmentation patterns of your experimental data and your CCS values with those predicted values or in silico values of the structure that it's predicted. Now, when we're talking about more quantitative workflows, we have another software tool called TASK. This is target and screening and quantification. And this is where you would set up your standard calibration curves and doing quantitative workflows in a more targeted way. But we also have recently in TASK implemented a flux model. So the significance of metabolic being on a slightly shorter time scale is going to give you a better picture of dynamic processes. So this is where you would take a labeled precursor using a stable isotope tracer such as carbon-13, nitrogen-15, or deuterium. You put that into your cell culture as a precursor, gets integrated within your metabolome, and then you get an idea of the overall flux or metabolism of that precursor into your metabolome. And you can set this at different time points to do throughout your experiment, or you can do a more steady state experiment where you take an initial versus a final full integration to look at what metabolic pathways are actually being triggered when you run your experiment and under different conditions. So how do we measure metabolic flux? Typically, you would use one of these LC-MS-based experiments. You incorporate it into your metabolome, and then you would employ the task FluxOmics module to look at how those labels have been integrated within your LC-MS data. We also include a natural abundance correction for naturally occurring isotopes. Then that will compensate for what you would naturally see versus what has been integrated from your labeled precursor. And this is CCS enabled as well. So one of the standard precursors that we would use that is labeled is fully labeled carbon-13 glucose. Now, there are a lot of isomers that are exactly the same mass as glucose, such as fructose. Now, we can actually separate those in ion mobility, and then you are not subject to your any interferences from isomeric or arsenic species when you're doing this type of experiment. So the way that we do this is it is based on a targeted method. So you would have to have the analytes of interest. You can base this on a previously developed quantitation or targeted workflow. You can also use various pathways that are known within literature. So this is what's known as the small molecule pathway database. So we have connectivity with that database, and we can pull in any metabolic pathway and build a method based on that metabolic pathway. Of course, if you have a list of analytes that you're interested in looking at on your own, you can put it in a CSV format and integrate that into your method. And this will automatically generate isotopologs that it's going to look for within your data analysis workflow. So this is an example of how it looks like when you do your data analysis. You get your quantitative results of isotope enrichment within your metabolome. This is amenable to large batches. It exports into flexible formats such as CSV or Excel. And this allows you to do further downstream analysis if you wish. And I will repeat what Shorjo said, our data formats are open formats. We do like you to be able to have access to the data and do further analyses as you wish. If you have bioinformaticians that want to have more complex workflows than what we have available. And we do have APIs within our both Metaboscape, and Task is also very amenable to being able to export formats that you can use in further bioinformatics workflows. I will give some props to our developers and our partners in Southern Denmark, Niels Spragerman, and a number of names that you might be familiar with. The group up in University of Wisconsin-Madison, Erica Pierce's group, has done a lot of work that has helped us out with developing these types of workflows. And now what I'm sure you all are looking forward to hearing, my next section is on single cell. We're focusing first on lipidomics. So towards our single cell lipidomics applications, it's because we tend to use SRM 1950 with lipidomics extracts more frequently. This is where we focused our development on for our single cell applications. So this all started with, again, this one paper where we were doing in-depth lipidomics analysis from limited sample amounts. So we wanted to try and move towards how low can we go to get extended coverage and more lipid annotations. And this is where we started with using the elute nano-LC, which is typically outside of my purview for the small molecule world. It's standard use in proteomics applications. But when you use a nano-LC and you couple that with a timsTOF Pro 2, you can get significantly more features being annotated because you get that nice sensitivity. Now, when we couple this with an SCP, and this is an example of a pharmaceutical cocktail mix in acetonitrile and water, and we've injected it both on a nano-LC with the timsTOF SCP, and we've compared this with our standard, ESI source and our timsTOF Pro 2 system. And I will call to attention that we've only injected 0.5 microliters of this mix onto the SCP and 5 microliters onto the timsTOF Pro 2. And the difference in signal intensity is astounding. We're looking at going from 1,000 to millions in signal intensity. And that's with a 10-fold difference in concentration level. Now, moving towards lipidomics, I should say that the swap from an HPLC to a nano-LC, not that complicated. I have taught many a grad student and even undergrads how to change sources. So our systems are very amenable to changing from one to another. And that's what I really like about these systems, is that they are easy to work with. So it's really just a change of a cap. We change from an ESI source to the captive spray source. You install the actual mounting of the captive spray source. For these lipidomics experiments that we've been running with our nano-LC and SCP for lipidomics, we use the ion optics columns just like you would for a proteomics application. Really easy to switch over. There are also some parameters that you need to change. I believe there's some multiple reference values that you need to change, and some deflector voltages that need to be adjusted. And we are in the works of setting those up as default methods for the SCP. So, and I totally agree with Shorjo with his statement that the SCP is a terrible name. It's like, I would like to name it the SCM, but I will never get my way with that one. So it really is our high sensitivity instrument. And it does not mean that you're limited to running it with proteomics. You can run this with metabolomics and lipidomics applications as well. So just to give you an example of a standard, our wonderful SRM 1950 extract and an equivalent of 3.3 times 10 to the minus 3 microliter equivalent of that extract on column. And we see a reasonably good separation. So this is very preliminary data still, and we can get nice looking runs using a nano-elute coupled with our SCP. And not significantly long run times, a little bit longer than we would run with our standard metabolomics and lipidomics workflows, but we're still looking at only a 30-minute run time for this gradient plus equilibration time. Now, to jump into some of the lipidomics results, I do want to point out that this is a very, very small volume that we're injecting onto this column, and we are seeing millions in counts for this particular LPC species. we do get a nice PASEF selection of our precursor, and we do see a fragmentation pattern that is indicative. This particular one on the LC side of things, coupled with the PASEF selection of the precursor and fragmentation pattern that is nice and clean. Ideally, I would like to see more fragmentation peaks. Lipid and our rule-based annotation and head groups and CCS, we do get good annotation on what these species actually are. And just to kind of finally point out, as an overview, the portfolio that Bruker has available for doing all of our different types of life sciences research experiments are available. And right today, we talked mostly about the timsTOF Pro 2 for our metabolomics and lipidomics applications. This is where the vast majority of our method development has been focused on. I touched briefly on our impact 2 with respect to the VIP HESI. This is basically this running a timsTOF Pro 2 in 3D mode. We also work a lot with our imaging group, which Kate is going to talk a lot about our imaging applications. So this is our timsTOF Flex instrument, where one half of the instrument is an ESI source and the other half is MALDI. Very easy to switch between the two, and we do have a lot of fantastic workflows that involve utilizing both sides of the instruments in our SpatialOMx workflow. And the front ends that we suggest for these types of experiments, mostly the Elute HPLC is our workhorse LC for our metabolomics and lipidomics experiments. And then we talked a lot about the SCP for our very low level analysis, moving towards single cell with our nano LC. Our target and screening quantitation workflows. That includes our Fluxomics module. And at that, I will finish up and thank everybody for your attention today. And